The overall goal of this procedure is to examine microtubule-based organelle transport in Drosophila S2 cells and primary neurons. This is accomplished by first suspending Drosophila S2 cells or Drosophila neuroblasts in cultured medium. The next steps are to plate the cells on Con-A-coated cover slips and depolymerize the actin filaments so that there is no organelle transport on them. Following this, fluorescent markers are used to visualize the organelles of interest, and time-lapse imaging is used to view organelle movement. Ultimately, results can quantify organelle motility through computational analysis.
Our research focus is in characterizing microtubule-dependent organelle transport by using high-resolution microscopy in Drosophila cultured cells. This video describes basic techniques of culturing Drosophila S2 cells and embryonic neurons and tracking organelle transport in these cells.
Drosophila genetics allows us to examine gene function in primary neurons by loss-of-function mutations, RNAi, or transgenic flies. This method was developed to extend our studies of organelle transport from S2 cells to primary neurons.
Begin by preparing the Con-A-coated cover slips. Place the cover slips in a ceramic rack and wash them by chromic acid immersion for an hour. Next, rinse the cover slips thoroughly with running water for 30 minutes until the acid is completely washed out. The cover slips should then be allowed to air dry.